This is my membrane transport tutorial, and in order to get the best out of this video, you'll need the worksheets which come from my website. So just follow this information here to go to zerobio.com, go to the metabolic processes strand, and download the worksheets, print them out, and then you can follow along. So this is for grade 12 biology, membrane transport. Okay, so if you turn to the first diagram, you're going to see this. And this illustrates the process of passive transport. Now, passive transport is the movement of molecules across the cell membrane. So here we see, right here, this is going to be the lipid bilayer, right? We've got a phosphate group right there, and we've got the lipid tail right there. And the phosphate bilayer conforms to the fluid mosaic model. It's fluid in that there are phospholipids here which are not rigidly attached to each other, not rigidly bonded, so that they can sort of move up and down. And there's also cholesterol, which is the 4 fused rings at various spots throughout the membrane, intercalating or pushing between the phospholipids, and that pushes the phospholipids apart. And that creates more fluidity. It's not a rigid structure. And also, some of the fatty acid tails on the phospholipids are unsaturated, and therefore they kink and they push the adjoining phospholipids aside as well. So this can happen throughout the membrane. So there's three ways that the cell membrane is fluid. And all of this can be regulated sort of dynamically. Cholesterol can be put in or removed. Phospholipids can be built that are saturated or unsaturated, and this kind of thing. So that's the fluid aspect. And then the mosaic aspect is that, well, first of all, the major building block of the cell membrane, of course, is the phospholipid. But also, there are various proteins. Now, I wouldn't recommend you circling all of these because we're going to be drawing over this, but I just want to highlight that there are many different types of proteins that are embedded in the cell membrane, either all the way through, as in these cases. So these would be integral proteins, but there can also be proteins just studying the surface here with receptors on them, carbohydrate chains to make receptor proteins, or recognition proteins, or of course adhesion proteins and that. So there's integral and peripheral proteins. So that makes up the fluid mosaic model. And now we're going to look at how molecules move through the membrane. And passive transport is the diffusion of molecules. So diffusion refers to having molecules in, say, a high concentration on one side of the membrane and a lower concentration of molecules on the other, and the molecules are going to move through the cell membrane like that, from a region of high concentration to a region of low concentration. And we are going to use these brackets over here to represent the concentration here. So the up arrow means high, and the down arrow over here means low. So we're going to go from a high concentration to a low concentration. In this case, we are going to be moving from the ECF outside. We're going to call the top part the ECF, and the bottom part we're going to call the ICF here. And we're going to move these molecules through the membrane. So I don't have anything in the bracket right here. I'm just going to put X to represent any of these molecules, and X here. So for all of these cases, there's going to be a high concentration on one side of the membrane, a lower concentration on the other, and in this case we're saying high concentration outside, lower concentration inside. But that it doesn't have to be that way. It could be high inside and low outside, and molecules could move out. All right, so let's go through the process of passive transport. Now, the first thing I'm going to get you to do before we really start labeling up this diagram is to copy down a note. I'm going to pull this down here, and I'm going to get you to write this down on another piece of paper or somewhere where you've got some room, because we're going to be drawing all over this. So you won't have room here. You might have a little bit of room here, but as you can see, I've got some other panels here. So passive transport is the diffusion down or with the concentration gradient. So a concentration gradient means a difference in concentration, and we're going to move down the concentration gradient or with the concentration gradient. In other words, we're going to move from high concentration to low. But keep in mind that just because that this says down here, it doesn't mean that we're always moving downwards. We could be moving this way, but if the concentration is high here and low here, it's still called down the concentration gradient. All right? No energy is needed for this process, so we'll talk more about that when we actually do need energy in the form of ATP. And this is for molecules involved with osmosis, simple diffusion, or facilitated diffusion. So these are the three processes here that are passive transport processes. So if you need to pause the video to write that down, go ahead and do that. All right, so we are going to look at the process of passive transport, osmosis, dealing with this molecule right here. So I've drawn this to look like a water molecule. So go ahead and label that water. And while we're at it, let's label this as oxygen, O2. Let's label this as sodium. And then all of these will represent either sodium, or this could be glucose if the concentration gradient is proper. We'll talk about that in a bit. So water moves through the cell membrane just like this. It goes right through the phospholipid bilayer from high concentration out here to low concentration here. So in this case, it's entering the cell down the concentration gradient, and this is a process of diffusion. It's a special case of diffusion, so that if we have a lot of water molecules out here and fewer water molecules in here, water will tend to enter down the concentration gradient by the process of osmosis, just a random movement bumping into the membrane and being able to come through. Now, the interesting thing about water is that although water is a polar molecule, like sodium is, all right, sodium being charged, water just being polar, water is still able to go right through the phospholipid bilayer. But as you can see, I've drawn this arrow here indicating that charged things like ions generally can't go through the cell membrane, although water can. So water is a special case. Now, this is osmosis, so I'm going to write osmosis down here for process 1. All right? Osmosis is the movement down a concentration gradient through a living membrane for water. All right, so I've got a little thing for you to write down here. Osmosis is the diffusion of water through a living membrane. It's moving down the concentration gradient, and again, no energy is required. This is passive transport. Let's move on to oxygen. As you should know, oxygen is a non-polar molecule, right? This is non-polar. And because it's non-polar, it is hydrophobic. All right, it is not attracted to water. It is attracted more to the hydrophobic fatty acid tails. Because of that, oxygen also can diffuse right through the cell membrane, and this is the same for other non-polar small molecules like methane or CO2. All of these non-polar molecules diffuse like water does right through the phospholipid bilayer by the process of diffusion, and so this is called simple diffusion. So let's write that down here, simple diffusion. So these two are very similar, just osmosis is a special case of diffusion for water. All right, so now we're going to move on to the sodium here. If we've got a charged ion, like I said, it can't go through the cell membrane just by regular diffusion. It needs to be assisted by membrane proteins, and these are integral proteins that span the membrane, go all the way through. So this would represent sodium here, and what's going to happen is sodium is going to travel through this open channel, and that's what we're actually going to call this down here. I'm going to call this an open channel, and this is a protein. All right, this channel is made of protein, and it's an integral protein. Channel's made of protein, and the sodium is going to move from a high concentration out here to a lower concentration in here. So it's moving down the concentration gradient from the ECF. Maybe we should write that. ECF out here, ICF here, in case you don't already have that. And the protein is going to go through. This is just a protein, sort of a tunnel that helps the charged ion, in this case, move through. Now, this could be glucose. That could be another way for glucose to enter, as long as it's moving down the concentration gradient from high to low. All right, because glucose has some charged regions on it. It's got those charged hydroxyl groups on it, so it can't go directly through the phospholipid bilayer. It needs to be helped through. So this is a type of facilitated diffusion. That's why I put number 3 here. All of these are types of facilitated diffusion, assisted diffusion essentially, and in this case we've got an open channel. Now this one is going to be a gated channel. Gated channel, and this part here represents the gate. So in this case, the ion, let's say, this could be sodium, could be potassium, chloride, whatever it is, or glucose even, is going to come down here, all right, and the gate is going to swing open, and then the molecule or the ion is going to enter that way. So it is facilitated diffusion, but this is the gated channel model of how it would enter. We also have this style here, and this is sort of the typical way that this is represented in physiology kind of textbooks, and this is going to be the carrier model. All right, I'm just going to call it carrier here. So again, this is a protein here, and it acts as a carrier. So the ion, let's imagine this is sodium, again moving from high concentration here. It's trying to get in to where it's in lower concentration, but this time the ion, the sodium in this case, is going to come along. It's going to bind. There's a little binding site here. All right, that will be specific to the sodium, and there'll be a different one for potassium and chloride and stuff like that. And then this protein here is going to change in shape. So let me write something about that. Okay, so right over here where I labeled this, change shape, or change conformation, change in conformation. And this change in conformation is going to be what moves the ion into the cell in this case, down the concentration gradient. But this does not take energy, all right? There's no ATP involved. It's movement down the concentration gradient. So these are 3 models or mechanisms of facilitated diffusion. So let's have a look at whatever notes I missed. Did I get that one? Yes, there's osmosis. There's this one now. That stuff's going to be in the way. Well, you can still see that one. Simple diffusion, therefore, is the diffusion of small nonpolar or hydrophobic molecules. All right, so this was our simple diffusion here for oxygen or CO2, methane, whatever. Small nonpolar hydrophobic molecules moved by simple diffusion right through the phospholipid bilayer. And I'm going to erase all that stuff, bring this down. And then facilitated diffusion is the diffusion of ions or larger polar molecules through membrane proteins. So for example, sodium or glucose, amino acids, all of these things moving by facilitated diffusion through integral proteins according to these models down the concentration gradient with no energy required. All right, I'm going to move on to the next diagram. And this is active transport. Now I've got two different models of active transport here. I've got something to do with this diagram and then both of these. So this is 4 for the hydrogen ion pump, and this is 5A and B for 2 states of the sodium glucose coupled channel, which is an example of cotransport or symport. For the hydrogen ion pump, I've called this uniport here because this will be transport of one thing in one direction. So let's write down the definition for that. Active transport is the diffusion up or against the concentration gradient. So we're not going down and with the concentration gradient anymore. We're going up or against the concentration gradient. So we're moving from low concentration to high. So against or up. Energy in the form of ATP is needed. Passive transport did not require energy. The following models here will require energy. And our examples are the hydrogen ion pump and or the sodium potassium pump, or just the hydrogen pump and the sodium potassium pump. So the first one is the hydrogen pump, and this is an example of uniport, transport of a single molecule in one direction. And symport will refer to the second model, which is transport in the same direction, but we're going to be moving more than one molecule. So we're going to be moving sodium ions and glucose molecules, but we're going to move them both in the same direction. So it's called symport as opposed to uniport, where you're going only in one direction. We're now moving two things in the same direction. All right, so I'll clarify that as we go along. All right, so let's have a look at the hydrogen pump or the hydrogen ion pump. So in this case, we've got these triangles here. So that's going to represent hydrogen. All right. And as you can see, I've got a high concentration of these hydrogen ions here in the ICF compared to the ECF, where I've actually drawn none of them. So I'm going to have a low concentration of H+. And these square brackets, of course, mean concentration, right? Whereas there's a high concentration on the inside of the cell. And what we're going to do here is we are going to move from the relatively low concentration outside to the higher concentration already present on the inside, as if we want to stockpile these, let's say. All right? So to move this hydrogen ion from outside here to inside, this is going to take energy to push it against the concentration gradient. So imagine that this triangle here, the hydrogen ion, is floating around out here. It comes in, it binds to a spot on this transport protein, but it can't get in because it's got to get pushed. And so ATP is going to be involved. That's going to give energy. And this circle here is going to represent a phosphate group. So I'll show you how that works in a second. But ATP is going to come along, bump into the pump, and come down here. And the ATP, adenosine triphosphate, will be converted to ADP, adenosine diphosphate, by giving up a phosphate group to the pump. All right, so it's called a pump because it's going to be pushing stuff. So ATP phosphorylates the pump, giving it a phosphate, going off as ADP, so we've used up some energy. That energy then is used to move the hydrogen ion in, causing a change in the shape of this, whatever the mechanism is exactly, but it will push the hydrogen ions in against their concentration gradient. And every time you want to take one in, you need to use an ATP. And that's how that's going to work. So this is going to be the hydrogen pump. And this is an example of active transport, moving molecules against the concentration gradient using energy. This is not passive transport, it's active transport. All right, so we've looked at the first model, and of course this is moving in just one direction straight through, so it's called uniport. Now, let's have a look at model number 5 for active transport, the sodium-glucose coupled channel. I'm using these circles again to represent sodium. So these are going to represent sodium. And as you can see, I've drawn a few of them out here. So there's a high concentration of sodium in this example outside of the cell. There's a lower concentration of sodium inside the cell. And this model works in 2 steps. We're going to first deal with sodium, and then we're going to deal with the glucose. All right? So we're going to move from a low concentration inside to a higher concentration of sodium outside of the cell. So we're going to have to push it through. So the sodium's going to come along. It's going to bind here, and then we're going to need some energy to push it out. So ATP is going to come along like this, just like we drew a moment ago. So let's draw an ATP here, going to ADP, phosphorylating the pump, giving it energy to push the sodium out. Whenever this happens, we use up an ATP and we start to build up the sodium concentration outside of the cell. Now, let's come over here. These structures here represent glucose. So I am just going to call this G for glucose. All right, you can write the whole word if you prefer. What we want to do in this model is we want to move glucose into the cell. And so let's say we want to stockpile it, eh? All right. Remember, glucose can polymerize to make glycogen. And so let's imagine we need a lot of glucose inside. But we've already got some in here, and we've got low glucose out here. And we've already got a lot in here, and we want more. We want to stockpile it, or we want to use it for cellular respiration or something. So outside, there's a low concentration of glucose. Inside, there's already a high concentration. So we're going to move it against the concentration gradient. This would be a good time to say down the concentration gradient. All right? So in order to move the glucose against the gradient, normally we would use ATP, right? Like we've seen in this model. But because this is coupled transport or co-transport, the sodium that we kicked out or pushed out in the first part now comes along and it binds to a membrane protein. And at the same time, glucose binds to the membrane protein. You need the 2 of them, like 2 keys in this very special lock. And to get the glucose to come in against its gradient, which would normally require energy, the energy from the sodium concentration gradient is what pulls the glucose in. Because the glucose can come and bind, and the cell will say, I'm sorry, you can't come in without energy. But because sodium is going anyhow down its concentration gradient, glucose is allowed to enter against its gradient. So it's kind of like sodium is driving in a car and it's going from point A to B, and glucose says, hey, can I hitch a ride? Normally I need to pay for gas myself, but the sodium says, well, I'm going there anyway, you just come on with me, all right? So that's how this works. So this is a coupled channel for the 2 of them, and it's co-transport. The energy for the movement of glucose against its concentration gradient comes from the movement of sodium down its concentration gradient, and that concentration gradient was established by actively pumping it out using ATP over here. All right? So let's write about that. The sodium-glucose coupled channel. Energy is used to build up the sodium concentration. So out here, let's say. Then sodium moves down its concentration gradient, allowing glucose to move up or against its concentration gradient. Okay, so even though I've drawn this coming downwards for glucose, it's moving up the concentration gradient, right? From low to high, or against the gradient. Now, if you were going to explain this on a test or exam, you wouldn't just write this definition. You would use all of these diagrams and a fuller explanation. This is just sort of a short form here. Okay, that then is active transport. I also want to talk about the sodium-potassium pump. Now, this is a very, very famous and very important model of active transport across the membrane. It is co-transport because two things are going to be moved, but it is an example of antiport. This is moving molecules or ions in opposite directions. So let's see what I've got about this for you to write down first before I show it to you. Antiport, transport of more than one molecule or ion or substance in opposite direction. One's going to be going one way, one's going to be going the other. And our typical example of this is the sodium-potassium pump. This is for two ions. Now remember, ions can't just travel through the phospholipid bilayer, right? They need to go through membrane channels or proteins or pumps or something like that. All right, so in this case, this is going to represent sodium, the little circle there. So that's going to represent sodium. So let's deal with sodium first. Sodium is going to be moving into the cell. So from the ECF out here to the ICF right there. And we are going to be pushing it against the concentration gradient. So there's already a high sodium concentration on the inside and a low concentration of sodium on the outside. Now we're going to deal with potassium in just a minute, and so that's what these brackets are for, but let's just leave it. So sodium's in low concentration out here. It's already in high concentration inside, and we want to move more of it in. So the sodium comes along, binds to the membrane protein, the pump, and in order to move it all the way in, we need energy in the form of ATP. So ATP comes along, phosphorylates the pump, the sodium-potassium pump, and is converted to ADP. That giving of energy by the phosphate group allows the entry of sodium. And it's 3 sodium ions that are pushed in against the gradient at the same time. All right? Now, let's turn our attention to potassium. Let's switch colors over here. These squares represent potassium, K plus ions, again needing membrane transport proteins to move through. And there is already a high concentration of potassium outside the cell and a lower concentration on the inside. So this is going to move against its concentration gradient because we're going to push it out. So there is high K plus already outside, low K plus on the inside. So the potassium comes along, attaches to the membrane protein, and you see we've got a phosphate group here. I'll switch over to blue. All right, we'll put a phosphate here. I'm just going to label the phosphate right there. I'm going to use a red, go back to red. When potassium binds, in order to push it out, the phosphate group has to fall off. The release of the phosphate group here, causes a change in shape, just as the phosphorylation caused a change in shape that pushed the sodium in. The release of the phosphorylation, that's called dephosphorylation. That causes a change in shape, which pushes the potassium out against its concentration gradient. And it's 2 K plus moving out. So what we have is the movement in of 3 sodiums and the movement out of 2 potassiums, all of these steps referring to the sodium-potassium pump. So I'm not going to write anything special down here. The whole thing is sodium-potassium pump. Okay, let me double check, make sure we got that. Okay. In case you missed any of them. The last thing that I want to talk about in this video is the concept of tonicity. And really, we're going to be looking at the movement of water. All right, we're going to be looking at the movement of water. That's an O there, not a 6. Let's get rid of that. H2O. Because, that's another 6. There we go. The movement of water. Because solutes, and that's what these stars represent. This is solute. Solutes cannot move freely, right? As we've seen, they're generally charged things, or they're too large. All right. So this solute, for example, usually when we talk about solute, we're talking about salts, things like sodium, potassium, that kind of thing. They can't move freely. They need facilitated diffusion, and that stuff is often regulated by gated channels and carriers and stuff like that. So water can move freely by osmosis. So if we take a red blood cell over here, and we're going to look at 3 scenarios, we're going to talk about the tonicity, which refers to the concentration of the solute, not really to the concentration of water. So in this case, you can see I've drawn a number of stars here representing solutes. So just imagine this is blood or something like that, all right? The red blood cell is going to have various solutes on the inside, and in the blood or in the ECF or in the solution around it, this is going to be the solution out here, all right, the part of the ECF, let's say. There's going to be a certain amount of solutes and a certain amount of water. An isotonic, iso for same, an isotonic solution means the solute concentration outside, I'm just going to use an S for that, is the same as the solute concentration on the inside. So I've tried to draw the same number of solute particles. If the solute concentration is the same, it means the water concentration is the same on both sides, and therefore, for the red blood cell, an equal amount of water will enter and exit the cell. All right, the chance of water entering is equal to the chance of water exiting. There's no net flow of water, and therefore the red blood cell maintains its shape and size. It doesn't swell, it doesn't shrink, and it is considered a healthy red blood cell. So this is called an isotonic solution, where there is equal concentration of solutes. That's what the tonicity refers to. But then the flip side of that is we can figure out the water concentration. If there's equal solutes, there's equal water. Now, let's look at another scenario here. If there is a higher concentration of solutes outside of the red blood cell, let's say in the blood, all right, let's say you ate a lot of salt, or you had a medical condition where you had lots of sodium or potassium or chloride or some solutes, some sort of solid particle in the liquid. There was a lot of it. Well, this is now called a hypertonic solution. It means there's a higher concentration of solutes outside and a lower concentration inside. Now, because of that, if there's high solute out here, there's going to be less water. If you were to take 1 milliliter of solution and there's lots of solute, there's less water. Compared to 1 milliliter of solution from the inside of the cell, there would be more water on the inside than outside. Now, don't get confused here, right? Red blood cells very small. The bloodstream is very big, right? So you're going to go, how can there be more water inside one cell than in all of the blood? We're not talking about that. We're talking about a similar volume, like 1 milliliter from inside compared to 1 milliliter from outside, just comparing concentrations. More salt outside. And because of that, because salt can't move freely through the membrane, I mean, it can move by facilitated diffusion or active transport, but all that stuff is relatively regulated, but water can just move very freely. Because of that, more water will leave the cell towards the salt. Water moves towards salt, you can put it that way. Or really what's happening is water is moving down the concentration gradient from high on the inside to low on the outside. Some water will make its way back in, but there is a net loss of water, and therefore this cell shrivels up. This is not an ideal physiological condition. This is bad, and this is called crenation, when the cell shrivels up and dies as it loses water. All right, so that's what happens when you have a hypertonic solution. There is a loss of water into the salty solution out here. Cell shrivels up, crenation. The reverse of that is when there is a high solute inside and a lower solute out here. Because we are talking solutes here, this is called hypo. Remember, hyper means high, hypo means low, but we're talking solute. So there's low solute outside. So outside we are going to have, where are we? Down arrow, out. Low solute outside, higher solute on the inside. As you can see, I've drawn it here. Therefore, on the outside, we are going to have higher water. These are always opposite. Lower, we're going to have, inside, we're going to have lower water. So high concentration of solutes on the inside means less water. Low concentration of solutes on the outside, let's say in your bloodstream, you don't have enough solutes in your bloodstream, you're going to have higher water. Looking at a comparative volume, let's say a concentration. And because of that, water again moves down the concentration gradient from high concentration out here to low concentration inside, or towards the solute. Less water is going to move out, and therefore, as water comes in, the cell is going to swell, and eventually, if there's enough water coming in, it's going to burst. This leads to cell death, and this is called cytolysis. Cytolysis, the splitting of the cell, the breaking of the cell. And again, this is not a good scenario for the red blood cell. So if you had tons of water out here, like you drank and drank and drank and drank tons and tons of water, water's going to start flooding in. This could cause your cells to swell and burst. So that's not good. Too much water is not good. Now, I just want to quickly mention over here that in a plant cell, because it's got a rigid cell wall on the outside, remember plant cell has this very large vacuole on the inside, and then the nucleus is over here. When there is a lot of water in the vacuole, this pushes the vacuole out against the cell wall. This is called turgor pressure. And this is actually necessary for plant cells, or beneficial. So you need to water your plants so that there's lots of water inside the vacuole, which will push it out and get a nice rigid cell wall. And of course, all these cells are connected together in a brickwork, and that gives some strength to the plant. Versus seeing this kind of thing where your plant or your flower is wilting because it's lost water, all right, and therefore the cells sort of collapse on themselves and they become flaccid. So high water pressure here in this case is important because of the cell wall, but in this case it's very dangerous. All right, so that is my summary on transport of materials through the cell membrane. I hope that helped. See you later.